Predicting how a stretch of DNA will control gene expression is one of the holy grails of computational biology. If we can accurately score regulatory elements from sequence alone, we can design custom enhancers for gene therapy, metabolic engineering, and synthetic biology without ever stepping into a wet lab. But there's a fundamental catch. Modern deep learning models that do this, like Enformer or DNABERT2, are black boxes. They might predict activity accurately, but they don't tell you why. And in reward modeling for sequence design, black boxes are dangerous. They're highly vulnerable to reward hacking, where optimization algorithms exploit spurious correlations in the model rather than learning real biological grammar. Meanwhile, large language models have revolutionized step-by-step reasoning, but feed them a raw string of ACGTs, and they completely fall apart. They lack the biological scaffolding to parse regulatory logic. So how do we get the predictive power of specialized genomic models, combined with the explicit mechanistic reasoning of LLMs? The bottleneck isn't reasoning capability, it's representation. When you hand an LLM a raw nucleotide sequence, it sees arbitrary tokens, not transcription factor binding sites, spacing constraints, or chromatin context. To fix this, the authors introduced a structured input format called the Regulatory Context Card, or RCC. Instead of feeding raw ACGT strings, RCC compiles each sequence into a deterministic, auditable schema. It runs the sequence through established bioinformatics tools like FIMO to scan against the JASPAR motif database, extracting transcription factor binding site locations, strand orientation, and match scores. It calculates global statistics like GC content, which serves as a practical proxy for CpG island density and chromatin accessibility. Then it derives higher-level grammar tags based on motif arrangements, capturing co-occurrence, clustering, and spacing hypotheses. Finally, it explicitly injects the experimental context, like the cell type. This transforms an opaque nucleotide string into a standardized, biologically grounded feature table that an LLM can actually reason over. But structured input alone isn't enough. You need to teach the model how to connect those features to regulatory activity. The researchers constructed CRE Reason Bench, a dataset of nearly 83,000 samples that pairs RCC prompts with step-by-step mechanistic rationales. Each rationale is synthesized by conditioning a frontier LLM on the RCC evidence, the observed activity level, and a strict instruction protocol that forces the model to reference specific motifs, evaluate grammar, and build a causal chain. This feeds into stage one of their training framework, a chain-of-thought generator. The model learns to output a structured reasoning trace, typically 7 steps, grounded in the RCC fields, and concludes with a discrete activity level. Crucially, they train this generator using format-only filtering. They don't filter for correctness against the ground truth label. Why? Because at inference time, the model will generate its own rationales. Training on noisy, self-generated traces ensures the downstream regressor learns to be robust to imperfect reasoning, bridging the distribution gap between training and deployment. Stage 2 is where the actual regression happens. Jointly optimizing language modeling and continuous regression sounds elegant, but it's a practical nightmare. Backpropagating regression gradients through long reasoning traces causes activation memory blowups, and the competing objectives create gradient conflicts that destabilize training. So they decouple it. After stage 1 generates rationales for the entire training set, they concatenate the RCC prompt with the self-generated reasoning trace, but explicitly strip out the final discrete conclusion. This representation is fed into a backbone LLM with trainable LoRA adapters. They extract the hidden state at a designated anchor position, specifically the last non-padding token, which empirically aggregates the model's final summary of the conditioning context. A lightweight projection head then maps this representation to a continuous activity score, optimized with a standard MSE loss. The design ensures the regressor never sees the raw label during training. It must learn to predict the continuous reward purely from the RCC evidence and the mechanistic reasoning trace. The results are striking. On enhancer prediction benchmarks across K562, SK and SH, and HepG2 cell types, R3LM matches or exceeds state-of-the-art specialized models in both Pearson correlation and RMSE. They evaluate this under 2 complementary settings: Oracle COTI, where the regressor consumes gold standard rationales, and generated COTI, where it consumes its own traces. Even under generated COTI, the model maintains high predictive accuracy, proving it can extract meaningful signal from partially correct mechanistic reasoning. To prove the rationale isn't just decorative, they run a critical shuffling ablation. At test time, they randomly permute the rationales across samples, while keeping the RCC prompts fixed. Performance plummets from a correlation of 0.82 down to near 0, confirming the regressor is genuinely conditioned on the reasoning trace, not just memorizing sequence features. They also walk through a concrete case study on a long-distance NBEAL2 enhancer in K562 cells. The model's reasoning trace explicitly identifies the critical GATA binding sites BS2 and BS3, links high GC content to open chromatin, and hypothesizes cofactor assembly with TAL1 and KLF1. This aligns perfectly with wet lab mutagenesis data, showing that disrupting those exact sites blocks enhancer activity. It's a transparent, falsifiable hypothesis generated entirely in silico. This work fundamentally shifts how we approach sequence-to-function prediction. By compiling DNA into structured, auditable biological representations and training models to reason through regulatory grammar before regressing to a continuous score, R3LM delivers both high-fidelity prediction and mechanistic interpretability. It transforms the reward model from a black box oracle into an evidence-grounded collaborator for biological design. As we push toward automated CRE engineering and generative genomics, models that can explain their predictions aren't just a nice-to-have, they're a necessity. Thanks for watching, and I'll see you in the next one.